Institute at Mount Sinai. And today I'm going to talk about a new method to study the change of microRNA messenger RNA interactions due to environmental exposure. So exposure to environmental chemicals during early development can increase the risk of developing breast cancer later in life. And it is well known that microRNA drive the development of many diseases, including cancer, regulating the gene expression level. So we want to understand how microRNA messenger RNA interaction change as an effect of chemical exposure condition. For this analysis, we are going to use an animal study to assess this effect. And we are going to consider three chemicals, the P, PB, and TCS, that are chemicals present in many products that we daily consume. So basically, some issues arose because of the small sample size of our data set. So we basically have microRNA expression and messenger RNA expression for a control group and different exposure conditions, so DP exposure, MPB, and TCS. And the sample size is like 20 samples per group, or even less than 20 samples. And the number of messenger RNAs after some pre-filtering is 7,500, while the microRNAs are 272. So this is kind of a tricky problem from a statistical point of view. So what we want to do here, and this can be applied to many other applications, so we want to learn this microRNA messenger RNA interaction for different groups jointly, simultaneously, by borrowing information across them, because we expect that some of the structure would be actually shared across different data sets. And also we want to include info from existing database, such as microRNA messenger RNA interaction database. There are many of them. So in this setting, so there are actually existing algorithms doing that. For example, for the joint estimation of multiple related networks, there are these nice works that rely on graphical Gaussian graphical models that basically rely on a penalized likelihood approach. The problem is that the performance of these methods really rely on the assumption of Gaussianity and linearity. And there are many methods integrating different kind of information from existing database, including Bayesian networks, consensus techniques. I mean, there are a wide literature on that. But the limitation of many existing algorithms, it's the linearity and Gaussianity assumption that are always often made. And when these are not met, usually the algorithms are really computationally expensive. So this is kind of the problem. So we previously developed two algorithms addressing these two problems. So the joint random forest that was published last year that can jointly estimate multiple networks from multiple related networks. And actually we demonstrated that this joint random forest can outperform the existing algorithm based on Gaussian graphical model. In the paper, you can see that we have been running a lot of synthetic data examples, and actually we outperformed those algorithms. And then there is IRFnet, that is also a random forest-based method for data integration from existing database. And this is a paper from 2015, and we actually showed that this integrative framework can outperform the original random forest for network construction. So those are the papers. The first one was published on JPR and the second one on bioinformatics. So here what we do is just to combine these two algorithms in order to estimate these networks for multiple exposure condition and integrate information from existing database. So we have basically, what we are going to do is that we have microRNA expression that is a function of, messenger RNA expression that is a function of the microRNAs. And for each condition, we are going to basically estimate a random forest tree. At each node, for each node, we basically prioritize microRNAs that have a similar sequence to that messenger RNA that is in the response variable. And we basically are going to, given the sequence similarity, we're going to consider a subset of predictors. And that is going to be the same across the tree ensemble. And those probabilities are computed based on target scan, that is a comprehensive database of microRNA messenger RNA interaction. I'm not going to go into details, but these details are in the paper. Anyway, we prioritize microRNA that have a similar sequence of the messenger RNA. So this is actually a way to lower the dimensionality of the problem. And then there is the JRF step, the joint learning. So basically, we need to choose the predictor that that we are going to use to split observation into smaller, the two groups. So this predictor is going to be the same across all the ensemble trees, and it is going to be chosen to maximize the summation, the weighted sum of decreasing node impurity across all trees. So basically, in order to select the best predictor, we are not going to refer only to one ensemble trees, but we are going to consider them all. So that if a microRNA is associated to a messenger RNA in multiple trees, it's going to be basically sampled more often. It's going to be used more often in the tree construction. So this is a way actually to borrow information, and we have been we demonstrated in the past with the 216 paper that this way can really borrow information and can estimate the common structure much better. So after that, we estimate the ensemble trees. We are going to rank interaction according to the importance score, the random forest importance score, and then we are going to set up the threshold for that using permutation techniques and just setting up an FDR cutoff for the importance score. So given the threshold, we basically get the interaction that are basically significant under our model, and basically we're going to derive those interactions. So we applied this method to learn this microRNA messenger RNA interaction based on our model. Actually, I didn't tell you before, but basically before doing this, applying this method, we basically ran a univariate test to see if the chemical affected the microRNA. So just to see just a t-test or a non-parametric test to understand if there was an effect of chemicals compared to control on the microRNA, and actually there was nothing significant. Also, we ran a correlation test, but at the same FDR cutoff that we used, nothing meaningful actually came out. Everything is in the paper anyway. And so basically what we did, we implemented our model, and the results are that the all three chemicals result in a loss of correlation of interaction compared to control. So basically those are the number of edges for each network. This is on the diagonal. So you see that in control we have like almost 7K interaction. Instead for DEP it's like less than half. And actually DEP is the chemical where we have a dramatic loss of interaction compared to control. So this is a plot showing the top microRNAs in the network based on control. And basically for each microRNA, we show the proportion of specific control-specific edges in blue, the proportion of chemical-specific edges in red, and the proportion of shared edges. So as you can see, there is a lot of control-specific as we expect. So this is proportional because it has been adjusted, divided by the total number of edges. So in these 10 microRNA actually are responsible for more than 85% of the edges in control network. Actually in DEP, we have these 3 microRNA that has a remarkable loss in connectivity compared to control, more than 90%. And actually we focused on those for further validation. So, for example, we did an enrichment analysis. So just we considered the messenger RNA connected to these 2 microRNA in control, but not in DEP, not in chemical, and derived the enrichment categories that basically contained like gland development, gland morphogenesis, mammary gland development. And actually the aim of the study was to assess if this chemical could affect mammary gland development. So this is actually a nice result because it means that those messenger RNA connected to this microRNA in control but not in DEP are involved in these pathways. But then we wanted to experimentally validate these results. We actually did an hypothesis. What we hypothesized is that DEP exposure impact on microRNA activity, then that impact on messenger RNA. Of course, this is an hypothesis, so we are not sure if this is the right direction. But what we validated it was to assess if DEP exposure, the chemical, affect microRNAs. And in order to do that, we consider MCF7 cell lines that are breast cancer human cell lines. And we basically got 3 replicates for control and for DEP. And those are basically the 3 replicates value, and the red diamond is the mean between these 3 dots, basically. So, and okay, we have only 3 replicates, but you see that there is a good separation between the 2. So we can basically say that DEP exposure have an impact on microRNA. And without this kind of network model, we're not able to estimate this. So as further validation, we want to validate the interaction between these microRNAs and the messenger RNA. So we are currently working on that. So basically, we proposed this new model that can jointly estimate multiple networks and integrate information from existing database. This actually model can be used in different applications, such as EQTL analysis, that might be performed for different tissues, including integrating information from existing database. It's well known that EQTLs are shared across tissues. Or co-expression networks might be estimated for proteomics and gene expression data, for example, and integrating information from protein-protein interaction database and so on. We have an ArcRAN package that it's available for you if you want to use the algorithm. Also, the other 2 algorithms are on CRAN. Thank you.
So you showed that there's less microRNA in the DEP setting, right? So is just like the less amount of microRNA the reason for the loss of connectivity, or is there something going on that, I don't know, prevents the microRNA-mRNA pairing or something?
Yeah, we don't know. Yeah, thanks for the question. So it's not that there are less microRNA, yet there are less interaction between microRNA and messenger RNA. So it's like we have a loss in connectivity, and actually also in cancer, many times we have a loss in connectivity compared to control. So it's like when in control, you have these processes that are involved, and then it's like you get a disease or you get chemical exposure, and those interactions just break. And yeah, so this is what.
But you did have higher CT values in the DEP setting, right? So there is less microRNA.
No, the number of microRNAs are the same. Yeah, more or less are the same, but are less interactions. The nodes of the networks are the same, but there are less edges.
Then what did you show with the difference in CT values?
This is the expression of microRNA, yeah.
Yeah, so you have less microRNA in the DEP setting.
Okay, you're talking about the values, yes. Yes, sorry.
So I have a related question. Your differences in CT are not much. You know, 25.3 versus 25.8. When I do qPCR, I'm looking for 10 versus 20. Something quite different. When you're in this area, especially when you're in the upper 20s, you're not really seeing much that's different. And I would contend that, in fact, you're not seeing much difference between the microRNAs, and that what's really going on is that there is something changing in the way the microRNAs are able to bind to their targets. Mm-hmm. What I would suggest is a Luciferase assay, where you build several reporters with some of the targets that you know about, and use these microRNAs in doses, but after treating those cells that you've created as reporter cells with your chemicals. That it wouldn't take too much, in fact, to do that. The transfection and the Luciferase assays are something a computational biologist can do. So
even if there is kind of a separation between the two?
It's actually not that hard.
Yeah.
Yeah, but you might want to consider the alternative hypothesis that, in fact, there's a little change but not much, and that change, what you're seeing here is, in fact, some sort of access to the microRNA change rather than presence or abundance of the microRNA change.
Thanks for your comment. Yeah.